So again, I'm Odedacia Foku, and I am going to talk about different soil processes that I can interrogate using these imaging techniques. And I will actually start with one of my favorite techniques, which is scanning electron microscopy. I've used this quite extensively in the last 12 years or so, and I'm not going to go into much detail because everybody talked about this different way how electrons interact with samples. But here is a cross image of electron microscope and a picture of electron microscope that we have in our laboratories. And in general, we have the electron beam that is interacting with a sample, and it generates a different type of electrons, second electrons, or characteristic X-rays. So here I'm going to talk about using some detectors that collect this kind of signals. So first I'm going to talk about SEM detector that is primarily focused on or collect secondary electrons and does surface analysis, surface sample topographies with high resolution at about 2 nanometers. Then I will talk about the energy dispersive X-ray spectroscopy, or EDX detector, that collects the characteristic X-rays, is used for chemical imaging, can give semi-quantitative data and also spatial maps. And I'm going to show them a little bit later on. And then finally, I will talk a little bit about FIB, FIB focus ion beam, scanning electron microscopy application. That method uses gallium ions, like Angela was describing before. They have high energy compared to electrons, so they are able to cut the samples and look at the interfaces between microbes and minerals, or between liquid phases that have been interact with minerals, so you can look at the reaction boundaries. Just giving a little bit detail on what is the speed of these techniques. So if we're doing single measurements at nanoscale or microscale, then a SEM technique is fairly quick. In minutes, we can collect these images. For the EDX, depending on the sample, depending on the concentration, we can be 5 to 30 minutes, depending if we're doing point analysis or maps. And for FIB-SEM, this is more challenging technique, and it requires more time, specifically for environmental samples, because they are probably one of the most challenging samples, since they contain soft material and hard material. So we can spend 4 to 8 hours depending on the type of sample. But we are living now in the time of automation, and we hope that automation will be a good approach for upscaling. So I'm giving here some different timelines, what happens to the automation if we use automation for this type of measurements. With automation, we have some new applications. One of them is called feature application. That gives us our opportunity to measure hundreds of particles at overnight in an overnight time scale. And these are probably some application that Swaroop is going to talk in a little bit. They're being used for atmospheric measurements and for particle count and particle measurements. Another application that I'm going to talk here about is something that's called large area map, or for short LAM application. This is getting the scaling up from micron scale to centimeter scale for EDX measurement. Imagine this like you can get a tile of 50 by 50 micron, or 30 by 30 micron, or 100 by 100 micron, and then repeat this tile across all your sample up to up to centimeter scale. And then you can create like a montage or like a puzzle, putting all these tiles together. Now you have a centimeter by centimeter scale for your sample. This takes about 6 to 10 hours. So again, quite a bit of time to be spent, but it will be much more time if we do it individually and sitting by microscope. And then I will talk lastly a little bit about this BX detector. This is a new detector that EDX companies that make energy dispersive X-ray spectroscopies have been developing. This detector, because of the geometry, has the ability to do these measurements I talked about in centimeter scale 3 times faster, so 2 to 3.5 hours instead of that. So we don't have the BX detector. I am trying to get one here in EMSL, and I hope that we can get it and do this kind of measurements here. Okay, so I am going to talk, just describe some of these examples. As I said, SEM for surface analysis, we can get a lot of information using a single technique if we're using a microscale region. For example, here we're looking at the transformation of a mineral. This is a work that's done in the lab. This mineral is calcite. You can you can tell that it's calcite by the cubic morphology. And when this mineral is exposed to organic matter, then you can see dissolution of the calcite and also formation of calcium organic matter association. So those, we can infer those just by looking at the morphology of the samples. If we have a sample that is coming from the field, and in this case, we have a volcanic glass sample that has been deployed in the field for about a year. Again, based on morphology alone, we can say a lot. We can infer a lot of what's happening in this sample. You can see some evidence of etching and tunneling in this sample from the fungal interaction. And we can see also the fungi hyphae just adhere to the samples. More information can get if we start putting the chemical imaging right now. So another example that I'm showing here is a quartz. We took a quartz mineral and we deployed it in the field for about a year. And then we take it out and we see this coating that is shown there in the circle. This is a region in interest. We're interested to know what that coating is. We put the chemical imaging, which is the DX detector, and now see that that coating is composed of iron. We will not be able to tell that unless we do this chemical imaging here. And then when we look also of the signal of the carbon into this area, then we see this very strongly association of iron and carbon. Again, we can tell a lot just by doing this kind of analysis in this scale. And then for the example that I showed before, when we put the chemical imaging, again, now we can tell more about what is happening around this fungal. We see these iron inclusions that are appearing on the surface of the mineral, and that gives us some clue about why this fungal hyphae might be in that region that we see it located right now. Also, I want to pay attention to this carbon map where it is very clearly, you can see where the hyphae is there based on the carbon signal. Also, you can see some hot spots of carbon around in that surface. So if we are interested more to know about it, then we can do the FIB analysis in that area that there is a rectangle in there. And based on FIB analysis, we can tell a lot more. So we can we can see some mineral dissolution just below where the fungi is located on the mineral surface. The fungi is depicted there with that dotted white line. We can see alteration layer that is happening in this region. We can see also inclusions of the iron inclusions that are shown in here with a different contrast. Putting everything together just gives us a lot more information of what is happening in that region, and specifically the fact that fungi is looking for nutrients and energy, and that's why it is co-located there with iron rich inclusions. So all this data and all this information is being gathered by using one technique alone. But now, as we have talked often here, one technique alone, it might not be enough to determine some very key important processes here, especially as Angela was talking about when we talk about the type of organic carbon that we have there. So we are now working with a few users, which is Rebecca Librand and Cassie Collins here, and also collaborating here with Dushan. You'll see an image where Dushan used his MALDI MS to give some information on the type of carbon that we have here. So we're using the SEM and the LAM detector. We can actually do analysis on about 18 different of these reference grids that we have loaded into that sample holder on the top. And we can do this overnight. We don't need to sit there by the microscope to do this type of analysis. And then if we are interested in the collocation, then we can look individually on these collocated fungi minerals and get some more information on how they are associated with a chemical composition of the minerals that are around there. And then, like I said, it would be nice to know what the type of organic carbon we have there. And although this is a different sample, this is like a proof of concept that we are developing right now. And we're developing this using MALDI, as I mentioned, with Dushan. And what you see in here, you see some quartz grains that have been deployed in the field for about a year, and then, excuse me, for about a month. And then when we extract those quartz, we look under MALDI, and we see for the composition of the organic carbon at the molecular scale, and we see the signal of this hexadecanoic acid. I think this is a lipid from what I can tell, and you can see this distribution of the lipid into the quartz. So that kind of information is super important for us. We need to know what organic compounds are and why they are there. So this is ongoing work, and we are continuing this now, moving from minerals that are pure minerals to the soil. So this brings me to characterization of soil structures. And here we are very interested specifically into the soil aggregates. There is an image here of the soil aggregate in the top. This is a SEM image. Soil aggregates are these intact structures that are reservoir of organic carbon. They have microbes, they have roots, they have organic carbon that has been absorbed on minerals. And they store this carbon at different stages of decomposition. They also contain pores at different sizes. So they are very complex. There are a lot of science gaps with respect to these aggregates. Specifically, the organic carbon kinetics of decomposition is established as a need to study these aggregates at the microscale and molecular scale. So how are we doing this characterization of aggregates? Tamas showed this a little bit earlier on, but if I go from left to right, we are starting with a macro aggregate. Everything that is above 250 micron by definition is a macro aggregate. And we're interested in organic carbon in that aggregate. So the first thing that we do is we stain that aggregate either with iodine gas or with osmium gas. One or the other will do. And after we stain that, we want to fix this aggregate, and we fix the aggregate in the resin. And the reason that we stain that earlier on is that because we want to separate the organic carbon that is in the aggregate versus this organic carbon that might be in the resin. After we do that part, then Tamas does the XCT analysis. He is able to determine the connected pores into the aggregate, the isolated pores in the aggregate, and the solid material that is there in the gray scale. And connected pores are in the blue, if you can see there. After we did that, we were curious if there was any changes in this aggregate as a result of resin impregnation. So we did some characterization before and after resin. We do see some changes, of course. We expect to see a little bit changes. They are not enough to change the chemical distribution around the pores. So after we do that part, then we take this aggregate that is impregnated in that epoxy, and we do these thin sections that are shown in that image there. And then every thin section is then put into the SEM, and then we do the chemical imaging using LAM application. That circle there is about 2.5 centimeters, just to tell you what the size that we're looking here right now. So the first thing I want to talk is about organic carbon. This is the soil macro aggregate that I showed there. What we did in this case, we did the chemical imaging. This particular aggregate is stained with iodine. And so then we look at for iodine. We know that iodine was specifically adsorbed on the organic carbon. So we look for iodine distribution and iodine signal in this map. And when we see the iodine, that will be used as a proxy for the organic carbon. And you can see in the figure on the right, you can see a lot of signals that are in red. Iodine here is a color in red. If you pay attention to those, and if you have ever done a cross-section of a root, though they look a lot like cross-section of the roots. We expect this. This sample was collected from Alaska coastal forest, is in topsoil. We have a lot of roots in there. So this is kind of convincing us that what we are actually staining with iodine, it is indeed the organic carbon. Another sample here is a sample with Devin Ripner, the first example with a collaboration with a user from University of Nebraska. This is with Devin. This aggregate, very different from the first one that I showed. This is a micro aggregate. This is about 250 micron. You can see now, again, the signal in the red that is organic carbon, organic matter, some form of decomposition. This sample was stained with osmium gas instead of iodine gas. Again, osmium here is in red, is used as a proxy for the organic carbon. You can see not only the larger organic matter decomposed in some form, but you can see some smaller contribution of organic carbon throughout this aggregate. So we are just being able to identify those, the carbon contribution into that. And I am putting back to this, the first aggregate, just to show how different those are depending when they're forming. The first one formed in the coastal forest. The second one formed in a dry land. It's important to look at those aggregates, and to Arunima was talking about using pore scale based from aggregates that are real in the soil. That shows importance because they are very, very different depending on the ecosystem when they're formed. So we are pretty sure that the organic carbon that was stained, what was stained was organic carbon, but we wanted to do some testing. So we used the number of minerals to see whether this organic carbon is going to adsorb, backing off. We wanted to see whether iodine or osmium was being adsorbed on other minerals in addition to the organic carbon. Here are a number of minerals that we used, and we used also some rocks. I'm not showing them here. The only, for the iodine, we see some uptake of the iodine on iron smectite. This is a non-tronite sample. It's not very widely distributed, but there are some areas that is distributed. We don't know why the uptake of iodine is in there, because it's very patchy and we have to figure out why it is in some areas and some areas not. When we do the same thing with osmium, again, those are soil minerals exposed to osmium. Now we see that this we don't see this the signal of osmium in the smectite. So I'm pretty sure that osmium is only staining the organic carbon. So going back to the 2D to 3D, you saw that video of the volume of the soil aggregates. We can take that volume, and we can do a number of segmentation, and we can create XCT digital images. We can create more than 1,700 digital images that are shown here. I'm showing 5 of them. Those 5 were selected for a specific reason, because they match very, very well 5 images that we all collected from the sectioning that we did of the aggregate. This is, again, EDX images. They look great. If you look at them, they look perfect matches if you look with your naked eye. But everybody that does machine learning tells us that there are some huge mortality challenges when we try to integrate those 2 different images. The first one is different resolution. The second one is that there are a lot of digital information into the thin sections that are manually done. And then we have one channel XCT, which means that we have only density-based map. And we have several channels on EDX, which means that we have different map for silica, or different map for iron, or different map for aluminum. They are distributed completely differently. So how are we dealing with this? We definitely are going through machine learning processes. This is with Maruti. He's going to talk a little bit about this. But in general, he is using random forest, which is a relatively quick technique that doesn't need a lot of parameterization. He does a match of the resolution between XCT and EDX. And then there is a co-registration of the images step. Then after that, a number of sub-images are being extracted. These sub-images are used through machine learning to predict the silica sub-image. And then if you compare this with the ground truth, it looks pretty good because that's what was used for training. Then we predict the entire silica section. And then we see that we are far from where we need to be. So Maruti tells me that we need to repeat this for 100 sub-images to get a good ground truth for one silica thin section. The other approach is with Peter Swart. He is going to talk about this. So I don't want to go here in details. He's going to cover that. But again, here he's using neural network for data fusion. It needs a lot more computing power to do this. And the goal, again, is you can use the 2D EDX data and the 3D XCT data. You can overlay those modalities, and you can get a three-dimensional with improved resolution. This is our goal. Like I said, Peter is doing a lot of this. I just don't want to go over it because he's going to go in detail. But no matter what process we're using, we see either resolution loss or information loss. So I want to finish my talk here, and I just this. Okay, so I want to finish with some of these challenges that we have. The integration of different chemical imaging is important because it does visualize these biogeochemical processes in a very challenging soil environment. It can identify the microscale processes that control macroscale observation. And lastly, it can address microscale to pore scale heterogeneities for prediction of soil carbon stability.